me? Good. Goping's pretty much covered my talk, so I'm just going to take questions. I have really become fascinated with interneurons, particularly the ones that reside in the brain or telencephalon. And I'm going to give you kind of an overview of that diversity, some sort of idea of where I think it comes from, a series of vignettes of how they form, and give you some flavor of where we hope to take our lab moving forward. So there's some sort of unwritten rule that you have to have Ramon y Cajal appear in any neuroscience talk, so I'm not going to break that. And if you actually read his works, E.G. Jones, who died just a year or two ago, translated as atlases, and they're a little on the verbose side. Here's an actual quote from him. The functional superiority of the human brain is intimately linked up with the prodigious abundance and unaccustomed wealth of the so-called neurons with short axons. It's a little verbose. You can shorten it. Basically what he was saying was, having compared brains from everything from mice up to humans, he was trying to puzzle the question of what makes humans special. On good days, at least, we like to believe we're smarter than mice. And the question is, what is functionally different about a human brain than a mouse brain or a rabbit brain? And what he concluded, for better or worse, was that what was really seemed different in humans was both the number and the elaboration of these local inhibitory cells. And he inferred from that that perhaps they were something special about them in consciousness. So if you look at the mosaic of cells within the brain and pull out the different subtypes, the cerebral cortex, hippocampus, and pallial structures are effectively made up of about 80% of these, the so-called principal cells, which are really long-range projection, excitatory glutamatergic cells, and this 20 to 30%, the 20% representing more what's in mouse and the 30% which is more what's in humans, of these interneurons. And so that sounds like a fairly substantial subpopulation, but these vary in abundance from the fast-spiking basket cell, which maybe makes 3 or 4%, down to some of the really more baroque rare classes, which are some fraction of 1%. And what Cajal hypothesized, and a lot of people have hypothesized since, is that this morphological diversity is reflective of functional diversity. And as we've learned more and more about them, it's really become clear that these have unique and interesting functions in the way you sculpt information in your brain. Kind of a simple way of thinking about information flow in the brain is that the first thing you do is take the external world and represent it internally, and that's really the domain of the excitatory cells. But to do anything interesting to them, you have to be able to attend, collect, and sculpt that information with great precision. And it's really these inhibitory cells that I think carry the weight of that. And as a result, increasing evidence, I won't say overwhelming yet, but I think it's coming, evidence suggests that when these guys dysfunction, brain dysfunction follows in close quarters. And so understanding how these guys function, how they integrate, and what goes wrong when they don't function correctly, I think will provide some real insights into what goes wrong in many people who have psychiatric conditions. So let's just take a kind of canonical cortical circuit and look at it for a minute and think of how information flows through the brain. Ascending sensory information comes up from mainly through the thalamus, somewhat directly from the olfactory bulb, goes into layer 4, is relayed up to the associational superficial layers, goes to other areas of the brain where presumably it's integrated with other information, eventually goes back to layer 2, 3, down to layer 4, 5, and then out. And so irrespective of whether you're looking at prefrontal, somatosensory, visual, or motor cortex, these basic layers exist in all those areas. And if you look at them, the other hallmark feature of this is that whether you look at the input, the output, the throughput, or the rhythms you see in areas, interneurons are really tightly integrated into these networks. And they're playing a role not only of shaping the information, but coordinating that information. And what we've hypothesized and really looked towards is how do they become integrated into the brain circuits, and how do they ultimately function in brain circuits. And while my lab is really more interested in the developmental questions, as I don't think I have to convince anyone here, really being able to genetically target and manipulate them is going to be key for really being able to understand their function in mature brain. So something we're all really familiar with are various forms of linkage studies where people find a gene that they're all very excited about and link it to some disease. And really one of the things from my point of view is what can I infer, what can anyone infer from a gene that truly is linked to a disease? And I think probably the easiest thing you can infer is that the gene, at least in the nervous system, often functions within the cell where it's needed. And so when we started thinking about genes and psychiatric disease, we of course were drawn to the question of, is there a Venn diagram overlap between genes involved in psychiatric disease and genes expressed in interneurons? So this is a beautiful kind of overlay of interneurons across the cortical layers done by Robin Tremblay from my collaborator Bernardo Rudi's lab. And we've, over the last 5 years, done a lot of RNA-seq analysis to look at the interneuron-specific gene expression and compared it to the other major player in the cortex, the pyramidal cells, the principal cells. I really, especially in this crowd, I was reading a review by Steve Hyman and Gopeng and Steve McCarroll, and one of the kind of admonishments to the field, they say, is, you know, there's a huge number of these linkage genes which become, as they put it, just so stories. And I have to say, if you're really hardcore about what genes cause which disease, a lot of that's loose, but you know, I think there's a real sense that we're getting more and more high fidelity, particularly with exome sequencing and private mutations, and getting a good idea of who does what. So what we found is if you assume the numbers may be wrong, but assume the ratios may be right, what we found was when we looked at the interneurons and just looked in a very broad way of genes that have been posited by any measure to be associated with psychiatric disease, we got a Venn diagram overlap of about 6%. And when we tried the same thing with the pyramidal cells, it was about half that. So, you know, again, I'm not sure anything particularly profound comes out of that, but we'd like to believe, and what I think will be borne out by further analysis, is that genes in interneurons may be particularly genes that, when mutated, are more likely to cause psychiatric disease. Or said in short form, when interneurons fail to function properly, psychiatric disease ensues. So here's some examples which I've kind of cherry-picked out, which are, I think, people have more confidence of, are involved in things like intellectual disability, Rett syndrome, or autism and epilepsy. And these are genes that are clearly, from our analysis, highly enriched in interneurons. Of course, the question is, they don't only function there, and so the kind of game we've played a lot and expect to play a lot more moving forward is the sort of thing where you damage these genes specifically in the cell types of interest and see if that causes the overt behavioral differences, changes that you'd expect to be associated with psychiatric disease. And while we've almost exclusively done this in mice, I have to say we're very much chomping at the bit to go to non-human primates and look at higher species, because I think one of the lessons, which again I don't have to convince this audience of, is that it's really hard to know when a mouse has autism. So moving from that general introduction to really the meat of the matter, we think a lot about interneuron diversity and how that comes about and how to tell one interneuron type from another. And you know, like many fields that involve butterfly collecting, you know, you get a lot of arguments of how many butterflies are around. And so there's the kind of minimalists, and I think I probably classify myself as more of a minimalist than an expansionist, are the lumpers who essentially want to say there are certain core features like morphology, connectivity, markers, or the intrinsic physiological properties that can pretty much tell you a class. But of course, there's the splitters who essentially say, let's turn a deli menu into this and make as many sandwiches as we can. So pick one out of the first column, one out of the second, one out of the third, and you have one of the 10,000 different interneuron types you could perhaps have. This is the kind of argument that goes on a lot. I'm actually not sure how profitable it is. when you think of cell types, at least in the nervous system, it's not clear to me that you necessarily need ones that can cluster apart, and that you need to actually put a number of 1, 10, 20, or 100 on there. It's possible that state changes between interneurons mesh them with actual class changes. And in fact, the best way for the brain to function is to cover all the space between them. And so we're really only kidding ourselves when we want to talk about different types. That said, the Allen Brain Institute just told us that in the cortex there are 23 different kinds of interneurons, so we'll go with that number. My point is, I don't really care how many there are. The thing I really want to know is, where does the information get imposed on the cells, and how does the information get imposed on the cells so that they do what they do? How do they get the certain shapes? How do they make the right axons? How do they choose the right afferent and efferent partners? How do they choose when and how to fire and their set point? And I think those are all discrete pieces of information that are tangible and discoverable. And what my lab really wants to understand is, how do you go from a pluripotent set of progenitors to these cells which arguably have exquisite and very precise connectivity and function, irrespective of where you look in the nervous system. So I'm going to start my talk off by very briefly giving what is kind of the gospel of my lab, which is an argument of where that information comes from. We divide that into what we call cardinal specification and definitive specification. And if I haven't already previously drilled this into your head before, I'll try doing it again. And essentially, it's not an argument of type so much as where we should look to get the right information to understand how the cells develop. And then I'm going to tell you 3 vignettes. The first one, really the part that's about genetic determination and genetic cell types. And I have to say, this has been an exciting, recently, an exciting collaboration both with Connie Sepko over at Harvard and more recently with Rula Satija, who was a postdoc with Aviv Regev. And I'll show you some unpublished and really exciting data that we've got from that. I'm then going to go on to what Go-Ping alluded to. One of the things we believe and what we found is that the interneurons, in all likelihood, don't have any a priori idea of where and what circuits they're going to go into. But that's puzzling, because if you take a brain, any brain, from almost any mature species, and look at CA1 cortex versus the hippocampus, other parts of the hippocampus versus the striatum, in each case, you can find a favored cell type and a favored connectivity with great precision. And if you don't know where you're going, the implication is you learn on the job, and we're very interested in how that learning occurs. And we believe that activity, particularly privileged neural activity in what I'd call a pre-critical period timing, turns on gene expression and alternative splicing to allow these cells to do what they're doing. And we're really starting to scratch the surface of how that works. Finally, I'm going to give you a vignette. This is work that's going to come out later this month or early next month on when we've started to look at the way these interneurons integrate into circuits. We've realized, we kind of made the assumption, or at least I did, that essentially it makes a rough topography and maybe it cleans that up a little, but essentially the first connectivity it does is equivalent to its adult connectivity. And when we've actually tested that assumption in a particular subtype of interneurons and really looked at its connectivity by combining rabies retrograde tracing. I see Ian Wickersham, who came up with this method when he was with Ed Callaway's lab, and then combined that with optogenetics, which lined up very nicely with kind of looking at their efferent function with optogenetics. What we've come to realize is that these interneurons actually have a transient developmental function, which seems to be distinct from the mature function. That's interesting from a point of view of how the brain functions. It's also, I think, really going to be important to understand how brain dysfunction occurs. We all recognize that a lot of developmental disease is developmental in its origin, and we kind of can assume that we can wait to the adult and see what's wrong, and that would be reflective of what happened early in development. But what this suggests is that there are these transient events that come and go and have a permanent effect much later on the nervous system. And so while you may think that all your defects are in fast spiking basket cells and prefrontal cortex, if you have schizophrenia, that's a hypothesis that's kind of out there in the literature. What I'll show you is that it has a special relationship with another interneuron type that's transient, But if that relationship doesn't occur properly, the fast spiking cell doesn't develop properly. So you may think it's all the fast spiking basket cell, but it may be who they talk to early in life. So I think that's a principle that's going to be important in terms of thinking about how the brain forms and what goes wrong in disease. So this is taking you back to the very beginning of my lab's interest in interneurons. What we knew from the work of Smart and Sturrock back in the 50s was that there are these strange bulges at the bottom of the lateral ventricle. So if you took a human brain at about 3 weeks of age, or a mouse brain at about 12 days, took a chunk of the cortex off and peered down into the lateral ventricle, what you'd see nestled at the bottom are these three eminences, the medial, the lateral, and the caudal ganglionic eminence. So they're there embryonically, and then by the time you get to birth in a human, or even close to birth in a mouse, they've disappeared. No one really understood what they gave rise to. And we, some years ago, started to use, actually originally kind of classical developmental biology fade mapping, but more recently genetic fade mapping, to really understand the relationship of these structures to the cell types that come out of them. And as you probably have guessed, or already knew, two of these three structures, the medial and the caudal ganglionic eminence, turn out to be the source of really all the interneurons, not just in the cortex, but in the hippocampus, the striatum, and the amygdala. And so it really puts a very strong link between the embryonic origin of these cells and the cell types you're going to find in the mature brain. And so the way we got into this was by taking a fluorescently labeled donor brain, cutting out our favorite areas, the LG, the CG, or the MG, dissociating them, and then using high-resolution ultrasound backscatter microscopy, essentially just use transplant methods to put them back in the brain where the genetically marked cells could be seen. This can be done without killing the animals, and then you can come back weeks later and see where they ended up. So these were alkaline phosphatase labeled cells. We've done it with EGFP more recently, which makes it more amenable to physiology. And essentially the lessons are pretty complex, but there's a simple take-home, which is that the LGE, the lateral ganglionic eminence, really doesn't make any interneurons in the cortex. It makes a lot of interneurons in the olfactory bulb, but mainly makes medium spiny cells in the striatum. Whereas the CGE and the MGE both give rise to large and diverse populations of interneurons. And as we dug into this and looked at it a little more, a simple lesson came out was that when you looked at the population that came from this structure and this structure, they were completely non-overlapping. And so this really was the first hint that site of origin, as it turns out time of origin, is a strong predictor to what kind of interneurons you're ultimately going to get, and really pointed us to the idea that if you wanted to understand interneuron diversity, that's something that starts right at the birth of these cells as they come out of these structures. So this is a Venn diagram kind of dividing up into the different interneurons, and it's kind of like a 2/3, 1/3 ratio. The green area are the interneurons that come from the CGE, and the purple area here surrounding the red and the blue area are the ones that come out of the MGE. And what we've said is that you can divide up the interneurons into three non-overlapping categories, CGE and then somatostatin and PV from the MGE. But if you actually get into it, there are a lot of subdivisions that occur in the CGE populations and more subdivisions that go within the somatostatin and PV cells. And so, while the origin gave you a hint of the diversity that occurred, it was really hard to account for the subtypes that ultimately showed up within any given region, like the somatosensory cortex, or even more pointedly, the interneurons as they differ in the cortex, the hippocampus, and the striatum. So it kind of suggested developmental genetics and imprinting on the progenitors at birth was important, but it suggested something was happening later to tell them more specifically what to become. And really that drove us to a question which we're puzzling on now and moving forward, which is how do you make these cell types which have general, as I'm going to call them, cardinal classes, and embed them in all sorts of different circuits which presumably do completely different things. So if you follow it from an embryonic point of view, the interneurons are born. They're born within these two structures where they proliferate. They migrate. They undergo this what's called long-range tangential migration, where they kind of follow the curvature of the brain to go to all the areas, cortex, striatum, and hippocampus, and amygdala. Then ultimately, they morphologically develop, make synapses, and mature. And they make these beautiful cell types and these beautiful connective patterns that have been well described in the literature over the last 10 years. This seems to be, as a first pass, a genetic program that's imposed on them, but as I've already alluded to, there are environmental cues, and those environmental cues are tied into neuronal activity. And one of the puzzles is exactly what is neuronal activity doing? Is it passive? Is it instructive? Is it simply a timing mechanism? And to be honest, I'll show you some evidence for it being important, but you could ask questions later. We really don't understand whether this is an active role of neuronal activity or just a passive role, which essentially, when they get to a certain point, activity comes online and that allows them to wake up and smell the signals around them that are going to tell them more specifically what they need to do. So we divide the interneuron development along the developmental axis into cardinal specification, which roughly speaking is those things determined at interneuronal birth. And those include things like their intrinsic firing properties, their efferent type tropism. What I mean by that, some interneurons like to make connections on other inhibitory cells. Others like to go to excitatory cells, some to the dendrites, some to the axon, some to the soma. And finally, there seems to be fundamental molecular markers which are immutable. So parvalbumin, for instance, is something that only turns on in fast spiking cells and not in other cell types, and that seems to be imposed on them right at the time they're born. By contrast, we have what we call definitive specification, which I say is determined at settling position. That's simply jargon for post-migration. So wherever they end up, what layer, what structure, what area, gives them really what I think is definitive specification, and that really is local connectivity. Who are they going to get connections from? Who are they going to make connections to? Their laminar and aerial positioning. And finally, which is kind of part and parcel of their connectivity, their dendritic and axonal targeting. Let me give you a quick example of a cardinal and a definitive specification, just so you know that I'm not speaking loosely. So here's a cardinal specification, what we call fast spiking basket cells. They're very characterized by being able to fire about 3 times faster than any other cell type in the cortex. They're parasomal targeting, and they're called basket cells because they form basket cells around principal cells and give large inhibition. And they have very typical markers like parvalbumin. However, here's a case where Robin Tremblay from Bernardo Rudi's lab has looked at basket cells in layer 5A versus 5B. These are literally cells that are no more than 20 or 30 microns from each other. And what you can see is the ones in 5A get input from POM and send their axons only to 5A, whereas the ones that settle down in 5B get input from VPM, totally ignore 5A, and give projections up to layer 4. So Paolo Arletta has had some beautiful work recently, which has kind of shown this in a more experimental way by showing that they have some intrinsic character. So if you put them in the wrong place, they can beckon forth the right cues, but also giving some indication that they also respond to where they end up in a specific way. Here's another example. One of our favorite cells are the somatostatin cells, which are in the hippocampus most famously called OLM. That means that they sit up in the Oriens layer and they send their projections down to lacunosum moleculaire. Their cousins in the cortex are called Martinotti cells. They generally sit in deep layers and send their axons superficially, most classically up to the layer 1. And then there's these low threshold spiking cells down in the striatum. Now, all these cell types, although they're in completely different structures, share a lot of commonalities. They're dendritic targeting, they're rebound bursting, they're facilitating when you excite them. And as we've been able to show along with Corey Harwell from Arnold Krickstein's lab, is that common lineages, in fact, give rise to interneurons that are destined to all these same 3 structures. So you can come from one lineage and go to these 3 structures. And I'll actually show you that data in a minute. So what is the evidence for a role of genetic determination in interneuron diversity? And as 2 extreme models, you can imagine that you have a clone of cells here, and they're going to form a column, a cluster, or a layer in the cortex, or maybe form a part of the CA1 hippocampus. Or alternatively, you can imagine that really lineage doesn't predict much, and they get scattered all over the place. And in fact, as I'll show you in a minute, the evidence really strongly suggests this. So that's interesting as an experimental finding, but I think as a functional finding, it's equally important because it tells you lineage does not specify their final position. And perhaps even cardinal specification is only telling you certain actual properties of it, but not actually dictating what structure in the brain they're actually going to give rise to. So this work was done by a wonderful postdoc in lab, Christian Mayer and Rachel Bandler. And what you see here are the green cells, which represent all the cells that you just put a retrovirus in and label. But using tricks that Connie Sepko has developed, we can put a barcode in so that we can recover the barcode and know with high fidelity who's related to who. So in principle, you might think anyone close to each other are linearly related. But when you look at the barcodes, you can see that this cell is related to this cell and this cell. And so that statement relies on statistical arguments. The best way I can say it in a simple way is when we take these pools of retrovirus and inject in 1, 2, and 3 brains and look for barcodes, we often find the same barcodes within one brain, but we almost never find the same barcodes in separate brains because the library is so complex that probabilistically, if you see the same barcode, it's very, very likely that the cell came from the same lineage. So in here, you might kind of infer lineage is important. They're all in the cortex. These two are beside each other. Maybe this guy got lost. But then when we started looking a little more, here's one where 2 of them ended up in the hippocampus and another 2 ended up in the cortex. You might again say, well, it's not really that far from each other, but there's a sulcus here. These guys must have they didn't swim across, so they probably got away from each other really quite a bit earlier in development. And then we saw a lot of stuff like this, which really kind of surprised us, where one's in the striatum, central striatum, another one's kind of on the edge of the striatum, and another lineage-related one is way up in the cortex. So if you do this in a more quantitative manner and just compare lineages, and each one of these lines represents a lineage, certainly in some cases you see clusters of related cells which are closely positioned together, but of course even these ones that are closely positioned have these guys that are quite a bit distant from each other. And then when you start putting it in terms of not only distance but structure, you can see that red, blue, and green represent cells found in different structures. And here, in probably the most extreme case, you have one here in the hippocampus, a couple in the cortex, and a few in the striatum. So from one lineage, you have cells crossing many, many structures. So at least at first pass, the rules clearly are not that lineage predicts where you're going. There could be a covert logic to this, which we're still trying to look at. It may be that as we bookkeep better, we'll see that there really are ones that cross structures and ones that don't. But I have to say at first pass, my guess is that while cardinal specification is very important in terms of lineage and birth date, actual positioning and wiring is something that's determined quite a bit later. And so that's something we're trying to figure out and chase. And so, the question is, if you have a common lineage that go out, how much will that position of origin predict morphology, connectivity, markers, and intrinsic properties? So, no better place to ask that question than here at the ground zero of single-cell RNA-seq, where Aviv Regev and Steve McCarroll and others have done a wonderful job of sorting these cells and looking at the lineages and really understanding the relationship between the expressome in a single cell and what they ultimately give rise to. So we've been very lucky in New York to get Aviv's star postdoc, Raul Statija, come and join us. And over the last few months, Raul and my lab, with the help of Chris and Rachel, have done a lot of work to try looking more rigorously at the developmental interneurons at different stages of development. And where we've really started is looking at those eminences. So we've started to take the MGE and the CGE, 2 areas we know interneurons come from, 2 areas that have a lot of commonality in gene expression, and start trying to ask, how do these diverse embryonic structures give rise to different cardinal cell types? So what you're looking at is about 2,000 cells just from the MGE. We have similar data, though it's really just in very early stages from CGE and LGE, where we've taken the cells and done drop-seq on them to analyze them. What you're looking at is the TSNE projection, which takes all the principal components and crushes that down into 2 dimensions. And then by looking at nearest neighbor analysis using the first 5 principal components, we're able to cluster these into different groups where each cluster presumably has near relatives that have something in common with them. And these are the kinds of patterns we're looking at when we look at individual genes. You can see that if you look at nestin, TOP2A, DLX, ARX, LHX6, or GAD1, all familiar players in terms of the early differentiation of these cells. Different parts of those clusters are preferentially labeled, and that lets us start to infer, as Rahul Satija had done in a beautiful paper with Aviv, pseudotime, where we can kind of make some guess of how they might progress here. And these are supervised, where we've kind of labeled the clusters. And you can imagine that the cells seem to kind of follow a pattern that looks roughly like this as they differentiate. So this is pseudotime because, of course, these are all cells at one point in time. But we're inferring through the nearest neighbor analysis is the logical progression of how they develop from a dividing pluripotent stem cell to a differentiated interneuron. We're actually doing this more longitudinally now, and this is kind of combining real time with pseudotime. And so this is where you can take them from different stages of development, bin them along this, and do PCA analysis to see how they cluster in terms of how genes relate to the various genetic diversity within that, and then start doing non-supervised tree building out algorithms to look at trajectories. And as has been beautifully pioneered elsewhere, but Rahul has helped us get into, this allows us, combining pseudotime and real time, to really infer that pattern of gene expression that goes from a cell to a differentiated cell integrated in a circuit. And what we hope to get out of this, and I'm going to loop back to this at the end, is we hope to really use this as a way to start figuring out how the genes progress and develop into differentiated structures. Am I really 15 minutes from the end here?
It's pseudo time.
20 minutes, okay. Okay, so I will, in real time, speed up a little here so I don't drag you out. Okay, so that's kind of the early developmental genetics. I'm going to take you through some very quick data where we've looked at neuronal activity, and here I'm crunching in a whole ton of experiments into a small number of figures, which really doesn't do entire justice to it, but lets you get a 10,000-foot overview in terms of the role of neuronal activity. And the last thing I talked about, which is this scaffolding transient connectivity that leads to adult development. So I've already shown you this picture, which kind of maps out neurogenesis, migration, morphology, and differentiation. And so we've become very interested in how activity might shape development. And so a simple way of doing that is by taking your favorite interneurons and either jacking up the gain or turning the gain down. And we can do that with a variety of ways, either by doing electroconvulsive shock or by using channels that either boost the activity or suppress the activity. And the simple answer is when you decrease activity, they get a lot simpler in their axonal and dendritic morphology. And when you increase the activity, they make more axons and more dendrites. So at least you can tweak their morphology and their development this way. In a set of experiments where we've really tried nailing this down in a more physiological context, we've looked at the neurogliaform cell, which we've genetically labeled, and we've asked, how is activity shaping its development? And so I'm going to really rush through a ton of data in a few slides and represents like a whole series of pharmacological genetic interference experiments in optogenetics. So the simple answer is the neurogliaform cell, when you reduce activity, doesn't like it so much, and it makes very small axons and dendrites. And when you start asking, where does that activity actually come from? Because obviously in context, activity comes from the afferent projections that impinge on that. And to do that, we'll label our neurogliaform cell with rabies, and then label who projects to it. And what we find developmentally is that they get a lot of cortical projections, we get a lot of subplate connections, and we get this big whopping projection from the thalamus going to these cells. And so we started saying, okay, that's who's giving excitatory input to these cells. What happens if we block that excitatory input in a selective way? So what happens if we take the cortex, and by blocking activity specifically in the cortex using tetanus toxin and genetic drivers that only block these cell types, what happens to the neurogliaform cell? And the answer is not much. It doesn't seem to care that much. So maybe that activity isn't so important. Well, we tried the same thing using thalamic blockers, and now we phenocopied exactly what we saw when we block the activity. So what it suggests is all activity is not created equal. Somehow the activity impinging on this cell from the thalamus is somehow functionally different from the activity impinging on this cell from the cortex. And so we started doing optogenetics, and basically now instead of blocking these cells, we put channel rhodopsin into the cortical cells projecting to this cell, and looked at the NMDA-AMPA ratios and got this kind of characteristic pattern, and then did the same thing from the thalamus. And indeed, both have NMDA and AMPA, but we noticed up here that there was a more pronounced NMDA occurrence from the thalamus, and even though I won't show you, using pharmacology, it turns out this NMDA represents a lot more of the NR2B, calcium more permeable form of it. So this is not only bigger NMDA, but probably indicating more calcium is going into the cell. So we kind of looked downstream of this and said, okay, something important in the NMDA receptor is going, and that's somehow leading through a calcium signaling, which calcineurin is often a part of, to the differentiation of the cell. And so we genetically started wiping out components of this, like one part of the NMDA channel or the other. And what we found is that having any interference with either of these channels or calcineurin had the morphological effect. But what was most striking, yeah, there you go, is when you block this, this is actually showing you back and forth what happens in terms of the connectivity. So this is the normal connectivity, but if we in any way interfere with the calcium signaling through the NMDA and then do the rabies tracing, what we found out is you lose a lot of thalamic input, and that is correspondingly reflected by an increase in cortical input. So what it's saying is activity is not only just important for the morphology of these cells, it's also important in helping to determine which afferents it's accepting connectivity for. So what I think my postdoc and student wanted to say is, oh, well, this is great, activity is instructive, and I think you should hold the press on that. It's in there, it's important, but in no way do we know it's instructive. And I can talk about that later, and I really think that's one of the challenges. We now know, at least in some contexts, that activity is important, but exactly what it's doing, we're not sure. I also want to add that beautiful work from Leach and Lowe and unpublished work from Chris McBain's lab, where they've tweaked AMPA and NMDA receptors and looked at connectivity of other cell types. Turns out we were really lucky in picking the neurogliaform cell, because many other cells, when you do these kinds of manipulations to them, don't seem to have any major change in connectivity or morphology. So again, is it a specialized case that we just got lucky and hit upon, or is activity always going to be important in the development of these cells? I'm saying, at least in interneurons, I think there's a more general rule, and I base that on the fact that as we've tweaked the other interneuron populations, and I showed you a bit of data on that, somatostatin and PV, it seems like activity has a generalized need for all of them. But exactly what it is something that really we're going to have to track down a lot more thoroughly. So this is beautiful work from Natalia DeMarco, who has her own lab up at Cornell, Theo Karianis, who's about to start his own lab in Zurich shortly, Rashi Priya, who just got her thesis, as well as Shebnem Tunç-Demir, and she's going on to a postdoc with Arturo Alvarez-Bulia, and she's just joined Renee Hand's lab. So at the functional end of this, we think that this really reflects two types of signaling, and I have a whole lot of data which I won't talk about, which I'm going to put under the purview of E to T and E to AS coupling. So what we know when we've started to look at how activity is affecting the cells is that there are a number of transcriptional targets that are activity regulated. This shouldn't come as a surprise to anyone who has been in the Boston area and listened to Mike Greenberg talk for the last 20 years about how activity changes gene expression. And so E to T coupling is going to be an important part of this. More recently, we find a lot of evidence of E to AS coupling, where alternative splicing of these through a number of specific splice factors, in our hands, probably most prominently RBFOX1, a gene that Douglas Black has done a lot of work on, suggests that alternative splicing is also regulated by activity. So in the last few minutes of talk, I'm going to give you that vignette, and which is, I think, kind of a warning to anyone who thinks you can look at the adult brain and infer what happened developmentally. And this is a story which Shebnem really formed the basis for her thesis, where she's shown that transient interneuron connectivity is required for adult cortical circuitry. So this starts off the same way as the other one. You do rabies, in this case targeting somatostatin Martinati cells, and look at their connectivity. And what you find developmentally is you get a whole lot of cortical input, but like the neurogliaform, a large thalamic input. And this was actually quite surprising, because work from a number of labs, Barry Connors most prominently, was surprising because the somatostatin cell in the adult doesn't seem to get much thalamic input. whereas the rabies suggested here it did get big input. So when we did that at P5 versus P18, we did discover that there were some differences, but many similarities, and here it's shown graphically. And the biggest difference is that while developmentally, the somatostatin cells in layer 5-6 get a big thalamic, ventral basal, and POM input, that input drops off by about 90% after you hit P9. So to make sure this wasn't an artifact of rabies tracing, I'm sorry, Ian, not everyone believes it, but I have to say in our hands, the rabies tracing has been very reliable. And so when we did the optogenetic approach, where we put chenorodopsin in the thalamus and then went up in the cortex and patched on to pyramidal cells and somatostatin neurons and did physiology at P6, as the rabies would predict, the green line shows you the kind of excitatory postsynaptic potential we got when we stimulated the thalamic fibers and patched onto a somatostatin cell. The black one shows the connectivity onto pyramidal cells. So there's something there, but it's a lot smaller. What's striking is by P9, this has reversed itself, and now the somatostatin input has got quite a bit smaller, and the pyramidal neuron, as you might expect from maturation, gets bigger. And if you go up to P18, it's much more dramatic. So really, this is what you'd expect: big thalamic input onto deep layers and layer 4 of the pyramidal neurons, and almost nothing onto the somatostatin neuron. So what about the output of this cell? So you can go down with optogenetics and look at the next link of this. And now you put chenorodopsin into the somatostatin neuron, shine light on it, and as you would expect, the somatostatin cell fires. And then you go look at the two cells that it could presumably connect with, the PV-expressing basket cell that's right sitting beside it, or the cortical pyramidal cell. And at P6, both of them seem to get connectivity from this. But what's striking is by P15, the PV interneuron gets an input, but a very small input, whereas the pyramidal neuron gets much bigger. So what this suggests is that there is a change in circuitry from P5 to the adult. So let's just pay attention to this one. The thalamus is projecting to the somatostatin interneuron, and it's then projecting forward to the pyramidal neuron and the PV cell. But after P9, this input has got much, much weaker, and now this cell is now only talking to this cell. So transiently, this guy gets thalamic input and talks to the PV cell, and by the time you get to the mature circuit, that circuit is pretty much gone. So, you know, if a tree falls in the forest and nothing comes of it, who cares? So the question is, does that transient circuit where thalamic information through somatostatin neuron to PV cell have any lasting effect on the circuits of the cortex? And in order to ask that, we did a simple experiment of going in now with diphtheria toxin and taking those normal somatostatin neurons and knocking them about into half by selectively injecting diphtheria toxin in the right cross into deep layers of cortex in a way that only the somatostatin cells die. And when we do that and then look at the thalamic to PV projection after P9, when it should be very strong, like you see here, it really is much, much weaker. So we've done this experiment in a number of different ways. I'm just giving one example. But the take-home seems to be, if the somatostatin neurons are damaged early on, the thalamic inputs to the PV cells don't develop normally. Now, you might say, well, that's great, but they're also dead later. Is this a developmental thing, or is this a permanent need? You did show me that there is a little bit of input from the somatostatin neuron to the PV cell. Maybe that's all that's important. So if you do the same experiment after P10 and do the same sort of recording, the thalamic to PV cell projection is just fine. So that's a very particular, very boutique sort of example, but I think it does illustrate what I think will be an important principle. That is that there are transient circuits that change during development, and at least based on this, those transient circuits are important to the adult, mature wiring in the brain. And what I think you can take home from that is the idea that developmental wiring is giving a scaffolding that is absolutely important for adult brain function. And if you want to understand adult brain function or how it might go wrong during development, you better study developmental patterning. So this work was done by Shebnam Tunç-Demir, helped by Bream Walmsley. So this is just a kind of retracing of the experiments I've shown you. I've showed you that genetic determination and lineage certainly predicts cardinal types, but not position within brain. I've showed a row of activity in terms of the morphological development of interneurons and their connectivity. And finally, I showed you evidence that transient scaffolding is important for the way the mature network forms. So what are the challenges moving forward? And I'm going to put that out there as a problem of genotype versus phenotype. We really want to understand how the genetic maturation and evolution of genes in these interneurons leads to their wiring in the normal brain, and conversely, how we might explain brain disease when those things don't occur properly. So one of the things we're really interested in is how early genetic cardinal specification versus activity-dependent factors affect interneuron development. A second thing we want to do is look at the Venn diagram of genes that are implicated in disease and understand how many of those genes, which hopefully, when validated, cause neurological disease, can be accounted for by their function in interneurons. And finally, I'm going to just show you a snippet of something that we're really excited about, with the idea that by understanding the genetics of different interneuron types, we can understand something about the DNA elements that will allow us to put things in vectors like viruses that allow us to target viruses so they're only expressed selectively in populations that we're interested in. And I'll show you a success story where we've taken an element, put it in an AAV, and now the AAV expresses highly selectively in interneuron populations. And it's something we think we can generalize to target any cell type you might want in the brain. So this is really the kind of hard work that Raoul is helping us with, looking at any given point in time at the selective gene expression in different areas, both at particular points in times and longitudinally, and to try figuring out how this gives you, in an unsupervised way, trajectories. And so you might see a branch point here during migration where gene expression along a given trajectory seems to split. And the inference is that there must be information changes that occur here that turn on those genes in those selective ways. And I think from a developmental biologist point of view, our challenge is to look at when those gene changes come on and use that to understand how environmental, intrinsic and extrinsic cues lead to the maturation of these interneurons. In terms of disease, there's two kinds of experiments which we've scratched the surface on, but I mean really just scratched the surface. And that is, how do specific genes affect interneuron function? So as we found your favorite disease gene, CHD8, which you're saying, man, that causes autism, we're confident of it. If we remove that or knocked it down, which is more like the human case, in just interneurons, would we get phenotypes that were indicative of an importance of that gene and that cell type? And I think that's one way of digging in and really connecting the gene to the cell type to the disease. The other kind of thing which we can get in there is by using optogenetic tricks and chemogenetic tricks, we can take our favorite cell types and turn the gain up and down to see how turning off specific cells in specific areas and specific times affects the way the circuits develop. So to put that in the context of interneurons, the MG interneurons, although really spread out throughout the cortex, really go into kind of layer 4, layer 5, 6 more prominently. And if you think of the cortex more like an engineer than a developmental biologist, the cortex is really divided into output layers, which are layer 5, 6, input layers, which are layer 4, and associational layers, which are up top. And the MG cells, at least disproportionately, have their role in the input-output layers. The CG interneurons, by contrast, are disproportionately in the associational layers. And so you can kind of imagine from an engineering point of view that CG interneurons might have more to do with association function in the brain, whereas the MGE interneurons might have more to do with the input-output functions of the brain. So to give you a taste of why I think that's reasonably tractable, in a conditional knockout where we've removed a gene called NKX2.1 from MGE and looked at the mice, this is a mouse where we've only knocked out about 30% of the cell's NKX. That has a very dramatic effect on the development of those interneurons. And you can see phenotypically this causes an ataxia. That's probably related to loss of the interneurons in the striatum, but also very clear frank epilepsy, which we can see when we do EEGs. And so this is a case where you're not knocking out all of them. You're really only knocking about 30% of the cells out, and you get a very dramatic phenotype. Let me give you another example, and this is of the CGE. And this is a bit of a cheat. What we've done is use a trick to get rid of cholinergic receptors on the CGE populations of interneurons. And this is, it's not in every case, it's partially penetrant, but it's a really striking phenotype where this ended up with this incredible, it moves pretty well, but boy, it really likes jumping in circles. I'm not sure how much that tells us, but it at least gives you the first hint of what we're trying to get at, which is, as we manipulate specific interneuron populations, how are they going to impact behavior and circuit tricks? Okay. Finally, I'm just going to give you a snippet of something that's working beautifully in the lab, and we hope to extrapolate. We've started playing around with various elements. Yes, we kind of hope to industrialize this, which is why we get wonderfully cryptic language like proprietary regulatory element. But anyway, when you stick this in an AAV, it's only about 500 base pairs. Put the AAV in the brain, you get a pattern that looks very much like interneurons. If you basically use this, which is just a genetic trick to mark all interneurons reliably, and compare it to what the virus expresses, there's about a 93% correspondence of, you get about 93% of the interneurons in the area of infection, and you get virtually no off-target expression. So here, you're taking an AAV. Trust me, I can guarantee you, we tried doing this with Cree, which doesn't work because even a whiff of Cree is enough to cause recombination in a reporter. But if you have something like EGFP, which is easier to see high versus low levels, it's clear that there's at least a couple orders of magnitude difference in the expression of this AAV in interneurons versus in pyramidal cells. So if you start putting chemogenetic tricks here, and now we've put the GQ Dread from Brian Roth in there, and then applied the ligand, we can see robust and high levels of firing in the interneurons when we patch onto it. And actually, I'm missing a slide, but what we've also been able to show, if you patch onto a pyramidal neuron and put the Dread on, even though that virus is presumably expressing there, you get no excitation, but you see a lot of inhibition, which we assume is from the interneurons infected around it. And I have to say, I've given this to a whole bunch of collaborators. This same virus works beautifully in ferret. There's some wonderful David Fitzpatrick data where he's been able to show orientation selectivity of interneurons in the ferret. This works in zebrafinch. Michael Long, a collaborator, has looked there. And we're now trying it with John Reynolds in primate and a number of other labs. So I think this is scratching the surface on what I think is going to be being able to take us out of just looking at mice and looking at non-human primates and other non-genetically amenable species to start looking at how manipulation of genes, so the same fun we've had for years in mice, can now go into other species and presumably humans. So this is just a thank you slide. I've really mentioned people as I went along, so I'll just leave it at that and take questions. Thank you very much.